All right. So good afternoon and welcome to the Northwest Pathogen Genomics Center of Excellence Pathogen Genomics Series. If you are joining us for the first time, my name is Stephanie Lunn and I go by Steph for short, and I also use she, her pronouns. Um, I am a bioinformatician on the molecular epi team here at WaDOH, and today we are going to be covering part 2 of the 3-part lecture series in viral genomics fundamentals. So previously we covered genome overview in our first lecture. In terms of today's lecture, we are going to be covering genome evolution. So there's some more assumptions of viral genome that I'll be talking about. I'll also review some evolutionary mechanisms and processes such as reassortment, recombination, mutations, some talk about selection and evolution as well. So I always like to go through my goals prior to beginning the presentation because this is basically the information that I hope to convey to everyone. So in terms of my goals today, I will begin by discussing how genetic variation can occur in viruses. We actually covered mechanisms of recombination in the 3rd lecture of bacterial genomics. So this time around, I'll be introducing reassortment and its impact on analysis. Next, we will talk about the mutation rate in viruses and how it can be quite difficult to quantify the mutation rate. Afterwards, I will discuss how genome sequences can allow us to infer an estimate of what is called the substitution rate, and we'll talk about that. I will also review how evolution can change viral populations and how viral genomes evolve in response to selective pressure. And lastly, give some examples of how viral genomes can adapt to host interactions. But first, let's start off with talking about genetic variation. And in viruses, genetic variation due to factors like mutation, recombination, and reassortment. To understand how organisms, including viruses, evolve over time, we need to recognize that genetic variation is essential. And genetic variation refers to the differences in genetic materials between individuals or populations within a species. For instance, genetic variation in humans could be things and include differences such as having 1 or 2 copies of the sickle cell anemia allele versus having none, or even having different blood types. For viruses, genetic variation arises through several mechanisms: mutations, recombination, and reassortment. Mutations are heritable changes in the base sequence of the genome. And for example, mutations in SARS-CoV-2 can affect its transmission, infectivity, and binding affinity to the ACE2 receptor. Recombination, on the other hand, involves the exchange of nucleotide sequences among different genomic molecules. And in the context of HIV, this can lead to multiple subtypes and circulating recombinant forms in different populations. And as I've mentioned, I previously reviewed recombination in more detail in the 3rd lecture of bacterial genomics fundamentals. And so those concepts apply here. So feel free to check out those lectures if you would like to learn more. And now reassortment. So reassortment is specific to segmented viruses, and typically their genomes might look something like this when illustrated. And it involves the exchange of entire RNA molecules between genetically related viruses. For example, in influenza, reassortment can create new flu strain subtypes. So understanding these mechanisms helps explain how genetic diversity is generated and can be maintained in both viral and host population. To talk a little bit more about reassortment, what is reassortment? So reassortment happens when 2 or more different viruses infect the same cell and exchange whole parts of their genomes. And in terms of how it works, it's usually when multiple segmented viruses infect one host cell. They create new viruses by mixing and matching genome segments from the parent viruses. And these new viruses or hybrids end up with a mix of genome segments from both parent viruses. So as a result, the new viruses can have genetic material from several parent strains. And depending on how the viruses package their genetic material, the result can be a complete mix of segments or distinct particles. So there is about roughly two types of viruses, just to simplify it, that undergo reassortment. Multi-part type viruses are viruses that package their genome in separate particles like bromoviridae or crisoviridae. And reassortment in this sense can create viruses with a random mix of segments from each parent. In contrast, single virion species, like influenza, for example, are viruses that package their entire genome in one particle. And reassortment in these cases usually leads to one virus replacing parts of its genome with those from another virus. And this process requires careful preservation of the signals and interaction needed to properly package the genome. So when discussing reassortment, it's crucial to mention influenza, which is especially well known for this process. New strains of influenza can emerge when human and avian influenza viruses reassort in other mammalian hosts like pigs. And in terms of how it works, usually when a single cell is co-infected with both human and avian influenza viruses, the segments of the RNA genome can mix and match. And the reassortment results in this hybrid virus with genome segments from both parent viruses, as you can see here. And specifically in influenza, this exchange can lead to changes in the H and N surface proteins, which are critical for how the virus interacts with the host. And it is worth noting that while certain H and N protein combinations are favored in avian hosts, both humans and avian influenza viruses can replicate very efficiently in pigs. And so this makes pigs an ideal host for mixed infection. Reassortment in these pigs can create new combinations of H and N segments, leading to new influenza viruses that can infect humans. And this process has actually been documented before, such as in Italy in the 1990s. And a notable example of this is the 2009 H1N1 influenza pandemic, which some of you might remember. The reassortment of human, avian, and swine influenza strains contributed to the outbreak, which was estimated to have infected a ton of people, somewhere between 43 million and 89 million people, and resulted in approximately 20,000 deaths. Let's talk about phylogenetic trees, because they can be very helpful when dealing with influenza. So phylogenetic trees show the hypothesized evolutionary relationships among species or other taxa. And a taxon is a group of organisms, such as a population, a species, or a larger group. And phylogenetic trees are a type of visual model that can take some practice to interpret properly given the underlying assumptions and caveats of the sequencing data that would be included in the tree. So one of the primary assumptions of phylogenetic trees is that they assume vertical transmission. That is, genetic material is passed from parent to offspring, much like parent virus to progeny virus. However, mechanisms like recombination and reassortment tend to violate this assumption because rather than genetic material being inherited from parent to child, genetic material is inherited through horizontal transmission, and that is usually among closely related species. So recombination and reassortment can invalidate phylogenetic trees, and typically this would reflect networks rather than a hierarchical clustering diagram. So if we were studying flu and knowing that flu strains undergo reassortment, what kind of visual models could we use to understand the evolutionary relationships? When reassortment occurs between influenza viruses, specifically in the HA and NA RNA segments, it can lead to incongruent phylogenetic relationships. And this is why visualizing the evolutionary history of these segments is so crucial for understanding viruses that undergo reassortment. And for this, we can use tangled trees, which are shown here, to visualize the evolutionary relationships between two surface proteins, hemagglutinin, shown here on the left, and neuraminidase shown here on the right. So these are two separate trees, one for HA and then one for NA. Now, tangled trees help illustrate the most closely related clades, showing that evolutionary relationships between reassorting viruses can actually be complex due to the differing relationships of the genome segments. So these trees are inferred separately and segments are matched by their sequence name. And this approach allows us to determine if the same clades are represented by both segments and how their evolutionary relationships compare. So the figure you see here depicts the outbreak from the 2017 to 2018 North American influenza season, which led to significant hospitalizations and deaths, more so than any year since I think the 2009 H1N1 pandemic. And this figure includes time-scaled phylogenetic tree. You can see time here on the x-axis. For the HA here on the left, and then the NA tree here on the right. And then the colors are going to indicate specific genotypes. So the trees connect the HA and NA sequences from virus isolates with the lines. And on the HA tree, branches are labeled by the HA clade. And while on the NA tree, branches are labeled by the neuraminidase amino acid substitution. And then the vaccine strains are going to be marked with the X's or the crosses. So looking at this tree, we can see that in this case, the closest relatives for the viruses with the A2RE genotype here in red on the HA tree are A2 viruses here in blue. However, their closest relatives on the NA tree are actually the A1 viruses in purple, which is very interesting. Moving forward, one of the things often talked about in the media, sometimes, I don't think we hear much about this anymore, but sometimes you might hear about viruses mutating, and viruses do possess a mutation rate. But what does that really mean? I've got a quote here, it's pretty long. It's about mutation rates that I really like. And it actually describes mutation rate as the probability of a change occurring in a genome sequence from one generation to the next. And several factors contribute to the mutation rate, including unrepaired DNA damage, errors made by the polymerases, things like intragenomic recombination events, movements of genetic material, and really any other molecular processes that introduce errors during genetic transmission. And we'll be delving deeper into mutation rates in the next few slides, but for now, it's important to understand that only the mutation and lineages that persist, often despite selective pressures, contribute to what we call the substitution rate. And this is what we measure through whole genome sequencing. So more details to follow on this very shortly. So the mutation rate of a virus is a crucial factor for understanding viral evolution and has practical implications as well. Accurately estimating these rates helps us understand how viruses evolve, but the methods for estimations can be varied and complex. And our knowledge of viral mutation rates is actually still incomplete, partly because measuring these rare and random events is challenging, and also due to things like confounding factors, biases, and inaccuracies. Additionally, the challenge of estimating mutation rates vary by virus types, which is linked to their different modes of replication. Some viruses undergo only one cycle of genome copying per infected cell, while others may undergo multiple cycles. And this variation contributes to uncertainties in mutation rate estimates. So a comprehensive review of viral mutation rates found that these rates differ among virus groups and are related to genome size. So on the graph, the mutation rate is plotted here on the y-axis as the number of substitution per nucleotide per generation, where a generation is defined as a cell infection. genome size is shown here on the x-axis and is scaled logarithmically. And you can see that the smaller genomes tend to have higher mutation rates. As we'll discuss further, single-stranded RNA viruses typically have higher mutation rates. DNA viruses, represented here, some here in this blue circles, they have lower rates compared to their RNA counterparts. Bacteria, represented by these black circles, have even lower mutation rates overall due to their larger genome size. So when we looked at how mutation rates vary among virus groups, we saw that RNA viruses have higher mutation rates. And this high mutation rate is a defining characteristic of RNA viruses, primarily due to their error-prone replication process, also known as low-fidelity replication. And what low-fidelity replication means is that the virus replication process has a high error rate, leading to numerous mistakes in new viral genomes. We'll discuss more about this in a moment, but essentially this error-prone replication is due to the polymerase enzyme being prone to making errors during replication. And so we see here in this top figure an RNA virus with low-fidelity replication. And over time, what this results is in a diverse population of viral genomes, as shown here on this right. However, when we sequence these viruses and generate a consensus sequence, which an example is shown right here, we really only capture the average sequence of the entire population. And I'll be talking about this a little bit more in detail in lecture 3. But given that RNA viruses are characterized by their high mutation rates, what are the practical implications? How does this affect the virus? So researchers have studied the distribution of fitness effects caused by single nucleotide substitutions in RNA viruses. And figure A illustrates the frequency of fitness values associated with random single nucleotide substitutions, while figure B shows the frequency of fitness values for mutations that have been observed and documented. So frequency is on the y-axis and fitness is on the x-axis. And fitness means the ability to live long enough to survive and reproduce and pass on those traits. A fitness value of 1 is basically the reference group. And what it means is that the organism reproduces or survives at a normal or average rate compared to others in the population. A fitness value less than 1 varies, with 0.0 meaning complete absence of fitness. The organism cannot survive or reproduce. 0.5 means it reproduces half the rate, indicating disadvantages in certain environments. And 0.9 means a minor disadvantage. A fitness value greater than 1 indicates a percentage where the organism is reproducing more effectively than the reference type, with 1.1 meaning 10% more effectively and 1.2 meaning 20% more effectively. So looking at these results, the results show that among all possible mutations, deleterious ones, and this is for RNA viruses, deleterious ones are the most common. However, in the actual viral population, deleterious mutations are still prevalent, but they are actually less common proportionally because some are eliminated by selection, usually purifying selection. So as a result, neutral alleles become more common and beneficial mutations remain rare, though they might be slightly more common proportionally. And we will discuss how these rare but beneficial mutations can increase in frequency within a population until they become fixed. But first, let's talk about the challenges of estimating the mutation rate. So as I mentioned earlier, estimating mutation rates involve varied and complex methods. And we really need to be cautious with these estimates because they can vary substantially depending on the data as well as the methods used. And so one major challenge in estimating mutation rates is that the error rates itself are difficult to quantify accurately. So to illustrate this, to try to be able to quantify the mutation rate, consider an analysis with something called a reporter gene. And a reporter gene can be used in experiments to track or measure gene expression as an indirect indicator of the activity of other genes or even regulatory sequences. And what it does is it provides us with a measurable product in experiments that allows us to monitor some biological processes. And the LAC-Z gene can be used as a reporter gene encoding beta-galactosidase, which breaks down specific substrates to produce a color change in gene expression studies in bacteria. So this gene is actually encoded in bacteria. But we can actually insert this reporter gene into a viral genome, such as HIV-1, and researchers can score mutations by inspecting the virus plaques. And this method seems relatively straightforward, but it can yield misleading data due to assay conditions and host editing of functions that are affecting the result. So what this figure shows, and you see here, this is the forward orientation or the plus strand, and this is the reverse orientation or the negative or minus strand. This figure shows that the errors in mutation incorporation are not uniformly distributed as each genome is copied. And the mutational profile for the orientation of each strand, forward and reverse, is also very different when you look at each of them. So this variability can lead to either overestimations or underestimations of the mutation rates. And then furthermore, experiments conducted in vitro revealed that the error rates of HIV-1 reverse transcriptase are extremely high and variable. And these errors can be attributed to several factors, such as alignment issues highlighted in orange, or primer strand slippage, highlighted here in the green. Misalignment usually occurs when the primer or the template strand is out of position. Really, they're not lining up. And this can result in improper base pairing. On the other hand, primer strand slippage usually occurs in repetitive sequences, like say you have AT, 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 and just a long stretch of that. And when primer strand slippage occurs, it can result in insertions or deletions of nucleotide. And lastly, some nucleotides are hotspots for mutation, which are marked by this asterisk, which I don't really think you all can see. But you can see the hotspots for the mutations vary between each of the strands across this gene, and this further complicates the estimation of the mutation rate. So estimating mutation rates can be quite challenging, if not nearly impossible, because it involves measuring errors during replication on a molecular level. However, we can use alternate estimates known as the substitution rate. And the substitution rate reflects the rate at which mutations accumulate over time and become fixed in a population. And this is observable through sequencing data. So to clarify the difference, mutation rate estimates estimate the amount of genetic diversity generated within a population of offspring, while substitution rates estimate the rate of evolution within a particular lineage or taxon. And substitution rates are generally well correlated with mutation rates, especially when we compare them among the Baltimore classification groups, which we have previously learned about. So when analyzing sequencing data, we typically estimate substitution rates rather than mutation rates. And this is because we are measuring the accumulation of mutations over time, not the error rates of the polymerase. And as shown here on the right, despite variations in these methods and computation, we can estimate the substitution rate for a pathogen to gauge the extent of divergence. Now, substitution rates are crucial to the concept of the molecular clock, a widely used method in phylogenetics. And the molecular clock hypothesis basically proposes that mutations or substitutions accumulate at roughly a constant rate over time in certain sequence regions. And by comparing the number of substitution between those sequences, we can estimate the time since those sequences shared a common ancestor. So molecular clocks are valuable because they allow us to incorporate time into our analysis, providing a chronological framework based on substitution rates. However, constructing phylogenetic trees, especially time-calibrated trees or time trees, is influenced by the evolutionary rate and the length of the sequence data used. And the evolutionary rate is specific, almost inherent to the organism or pathogen, while the length of the sequence data, whether it's specific genes, loci, or entire genomes, this can affect the estimated molecular clock and the temporal resolution of the analysis. So different types of sequence data, such as single genes versus whole genomes, can yield varying estimates of the molecular clock. Single genes often evolve at different rates compared to whole genomes, and they only represent just a fraction of the genome. And this can sometimes result in a loss of phylogenetic signal, making single gene trees less informative for certain analyses. Whole genomes, however, generally provide greater resolution and more comprehensive insights into evolutionary processes. So in practice, the choice of sequencing data, whether single genes or whole genomes, affects the phylogenetic analysis, and temporal resolution, or the ability to observe mutations over time, vary between single gene and whole genome approaches, as you can kind of see here in this figure. So according to the authors of this figure, the mean waiting time for observing mutation depends on the evolutionary rate and the length of the sequences. The green bands are mean waiting times of a month or less, and the pink bands are mean waiting times of more than a month. So whole genome data typically offers better temporal resolution, often detecting mutations about once a month, whereas subgenomic fragments may reveal mutations less frequently. And throughout the course of these lectures, I have basically talked a little bit about evolution, and viruses, in particular, viral genomes, tend to evolve in response to selective pressure. So a lot of what we have discussed about virus evolution might raise the question, well, what is evolution? So before diving into how viral genomes evolve in response to selective pressure, let's first clarify the concept of evolution. And evolution, in its simplest terms, is really change over time in a population. It is the accumulated change observed over a specified period happening at the populational level rather than the individual level. So to take a look at this figure, which illustrates the evolution of HIV in response to AZT, an antiretroviral drug. And AZT functions as a thymidine analog. And a thymidine analog is a chemical compound that mimics the structure of thymidine, one of the nucleosides, so basically a thymine base and a sugar. And it can be incorporated during DNA replication, but it often has modifications that alter DNA synthesis. But since it resembles thymidine, it can be mistakenly incorporated during DNA synthesis. And once incorporated, it can block DNA synthesis and introduce errors, leading to faulty replication. As an antiviral treatment, it inhibits HIV's reverse transcriptase, the enzyme that the virus uses to replicate its RNA into DNA. So initially, the population of HIV, shown here in the red, is susceptible to AZT, meaning that the drug efficiently and effectively inhibits the virus. However, errors during reverse transcription lead to genetic variability among the viral population. And some of these variants may acquire mutations that confer partial resistance to AZT. And this shift is illustrated as the population changes from red to green, where some variants can partially resist AZT, but not entirely overcome it. So as treatment continues, additional mutations may occur that confer full resistance to AZT. And these advantageous mutations allow the virus to thrive despite the presence of the drug. Consequently, the population evolves to become fully resistant to AZT, as seen here in this blue population, where none of the variants are susceptible to AZT. And this example shows how the virus has evolved from being AZT susceptible to highly resistant over time, reflecting a shift in the population composition due to selective pressure from the antiretroviral drug. And this ties into our next discussion on how viruses face selective pressures in their environment, with this scenario of AZT serving as a specific example of such selective pressure. So natural selection is an evolutionary mechanism by which individuals with traits better suited to the environment are more likely to survive long enough to reproduce and pass on those traits. Different types of selection affect the frequency of a trait in an environment and impacts the variation that exists in that environment. So discussing and reviewing selection is a whole nother lecture on its own. So in the interest of time, we will only talk about positive selection and its impact on viral genomes. So viral genomes evolve in response to selective pressure, and the host environment, which is constantly changing, acts as a form of selective pressure. For example, the use of AZT to target HIV infection is a clear instance of selective pressure. Mutations that confer a benefit to the virus, particularly in a specific host environment or niche, can enhance its fitness. And over time, these advantageous viral lineages may increase in frequency. So figure 1A displays windows of inferred positive selection for different beta coronavirus species, and you can kind of see that they're different. So positive selection refers to the process where beneficial traits become more prevalent in the population, really driving this sort of adaptive evolution. And this figure shows several signals of positive selection, each unique to the specific species represented by the red dots, and then others that are shared across multiple species. And then on the left, you'll see the phylogenetic tree of the beta coronaviruses annotated with the host species each primarily infects. And in general, the distribution of positive selection tends to be more similar among closely related species than among divergent ones. So figure 1B shows the genome-wide distribution of positive selection, highlighting segments of the genome that are under selective pressure from the host environment. And when we say that a gene is under positive selection, it means that mutations in that gene are favored by natural selection because it provides an advantage to the virus. And this is clearly illustrated here in 1B. And this leads us to our next discussion point, which is that viral genomes adapt to host interactions. And let's stick with SARS-CoV-2 as an example, because we have learned so much about this virus over the course of the pandemic. So over time, the changes that occur in the viral genome due to host interaction can lead to viral adaptations. And this means the virus becomes better suited to its environment. However, it is important to note that not all genomic changes are adaptive. And adaptations that are beneficial at one point in time may no longer be advantageous if the host environment changes significantly. For example, throughout the SARS-CoV-2 pandemic, we have observed numerous changes in the viral genome with each new variant. While the spike protein coding region is not the only part of the virus that has accumulated mutation, much of the focus has been on this protein, which is shown here in the circle. So I have circled the spike protein. So the spike protein is crucial because it interacts with the host cell, specifically binding to the ACE2 receptor to facilitate viral entry. And changes in the spike protein can affect its binding affinity, how well it binds to the ACE2 receptor. And sometimes these changes are beneficial to the virus, sometimes they're not. Another aspect of host interaction that has influenced the spike protein is the presence of antibodies. And some antibodies are designed to bind to specific regions of the spike protein or epitopes to prevent the virus from binding to the ACE2 receptor. And over time, the virus has acquired mutations that has helped it to evade these antibodies. Mutations that successfully confer resistance to antibodies can increase the virus fitness, leading to their spread in the population. And we can observe the impact of the mutations in the receptor binding domain of SARS-CoV-2, where they contribute to antibody escape. And this demonstrates how the virus is adapting to its environment, specifically to the selective pressures imposed by immune responses. All right, so that is actually it for today. Here are some discussion questions that you should be able to answer having sat through the lecture, and feel free to go back and review the recording and then try to answer them on your own. So something you should be able to do is give examples of how genetic variation might occur in viruses. How does reassortment in viruses impact phylogenetic analysis? You can talk about flu, for example. Why is it difficult to quantify the mutation rate? How can genome sequences allow us to infer an estimate of the substitution rate? What is evolution? What are some examples of how viral genomes can evolve in response to selective pressure? And what are some examples of how viral genomes can adapt to host interaction? So there were a lot of resources, a lot of studies that I had to pull from to make these slides. So here is the first set, and then here is the second set. And then with that, we'll conclude the lecture. Of course, you can contact me at any time if you have any questions, or if you're interested in the molecular epidemiology program, you can also give us an email here.